Okay, so okay, so it's a welcome to all of you on a very special Wednesday. We have in sort of audience all the participants of the special DBS at 60, which is a celebration of the department, its legendary history, and we have members of Department of Biological Sciences from the past. Some of them are amidst us. Of course, we are missing some very legendary figures, but that's okay. Now, in their all honor, we're celebrating this moment. So it's my great pleasure to have this colloquium within the context of this celebration. So thank you to the department, Maitreyi, and all people who have organized Amitesh and Devashish and Mahendra. I'm sure I'm missing some names, but all of you who have put your effort together in organizing this and bringing our expert speaker today, Renee Borges from Indian Institute of Science, thank you all for doing that. Of course, before the colloquium, we had some pastries, and thank you, DBS, for doing that. Yes, we had some pastries for the students. And as part of the rich tradition of the colloquium, I just want to mention, for those students, especially the biology students who are attending it for the first time, DBS from the past, Vijay, right there, sitting right there in the backside, would tell you that this series has been exemplary in showing works of natural sciences in this institute, which includes physics, chemistry, and biology. So, and it has built leaders through this kind of a series because you become much more broad-minded in length scales. As biology is saying, it's biology across length scales, or scales, essentially, in time scales as well. So you become much more broad. So I urge and I hope that the new DBS students who have just joined the department, a wonderful department to join, they actually recognize and exploit this forum much more and sort of participate in it so that they grow as a scholar. So thank you for joining in. So without further ado, I would let Professor Maitreyi Narasimha, who is my colleague, dear colleague from DBS, who is going to introduce our speaker today, Professor Renee Borges.
Thank you, Jyotishman. So, it's a pleasure to have Professor Renee Borges to deliver this Wednesday colloquium as part of the celebrations of 60 years since the founding of our department. I'd like to start by saying that she is at the Center for Ecological Sciences at IISc, where she's been since 1997. And her lab has been interested in studying associations or interactions between species on leaves, flowers, inside fruits, in the Western Ghats, in the Northeast, and also on this beautiful campus of IISc. And that's and she has sort of defined these sort of microcosms in which interactions between species takes place as ways by which to understand constraints that govern ecosystem health and that sort of thing. She's a native of Bombay. She did her undergraduate degree at St. Xavier's, her master's at the Institute of Science, not far away from here, and then worked on Malabar giant squirrels for her PhD at the University of Miami in Florida, and came back to India first to the Wildlife Institute in Dehradun, but was also director of research at the Bombay Natural History Society, again, not too far away from here before she went to the Center for Ecological Sciences at IISc. You'll hear a lot about interesting interactions between species. I want to say she's a very distinguished scientist. She's a fellow of the Indian Academy of Sciences and the Indian National Science Academy, and has also recently become General Secretary of the International Union of Biological Sciences, and has been awarded for her excellence in research by the Alumni Association of the Indian Institute of Science. So I would just like, she's also a J.C. Bose National Fellow now, and I'd like to thank her for accepting our invitation to come and speak at our meeting and to give this colloquium.
Thank you very much for that very generous introduction. And I honestly don't know why I'm here, but thank you anyway. It gave me an opportunity to come back to a city that I truly love and I have lived for many, many years of my life. This morning, I was delighted to hear people talk about molecules that they love. So I heard somebody talk about a complex that he loves. And to use that word love, you know, it shows passion. And all through the morning, I've seen how people have been so passionate and so excited about the work that they do, which they have communicated very well. And even when I had the opportunity to look at a few posters, I'm sorry I couldn't see all of them. But again, this same passion and this same excitement came through. And it's really wonderful to be amongst all of you and maybe to try and share some of my excitement and some of the things that drive me to do what I do. So I'm talking about life in and around microcosms. We know that scientists have been trying to simulate ecosystems, which is a very difficult thing to do. This was one such attempt in Arizona, Biosphere 1, which didn't do too well, Biosphere 2, which went on for a while, but again, extraordinarily difficult to control and maintain whole ecosystems within these structures. So people have then resorted to mesocosms, and we have all sorts of mesocosms, from ecotrons in Montpellier to autoponds in the Netherlands to oceanic planktotrons to earthtrons in Japan, where they're trying to simulate natural rhizocosms, and then limnotrons, where they have used recycled fermentation vats from a convenient beer brewery at the University of Guelph. And of course, most of us, I think, are familiar with microcosms that we artificially create. Of course, here's Richard Lenski very famously shuddering that, you know, all his long-term evolution studies might be smashed to smithereens. So these are the kinds of microcosms that we, you know, try to put together. There are many microcosms that are iconic and that are also created by joint efforts. In this case, the light organ of this squid has been created, forms a microcosm by bioluminescent Vibrio bacteria. You also have iconic microcosms in the pitcher plants. I think all of us are likely to be familiar or have heard of pitcher plants. And they have huge number of biota present in them. And interestingly, if you take pitcher plants across continents, you get very similar microbial communities in them, which shows that, yes, there is method in this madness. There is a lot of convergence, and it helps, therefore, to study microcosms across many different scales. So usually we talk about a macrocosm. A lot of us are also engaged in studying macrocosms. And then when we zoom in, we come to these microcosms. Here is a microcosm of a bromeliad, which has so many unusual and adapted organisms which are not found outside these microcosms. Special species of mosquitoes, for example, that live their entire life within the phytotelmata, which is the hollow water-filled cavity created by the bromeliad.
Renee, just a quick question.
Yes.
Just stopping you in the initial slides, but just a quick question for a broader audience. Can you just give a little bit of a length scale difference between the macrocosm and the microcosm?
A length scale.
Or what differences are there?
I think as I come along, you will understand it. So I'm talking about a microcosm as being anything that is self-contained.
Okay.
It could be at many scales.
Okay.
Okay, so it
Okay.
isn't, I mean, I can't give it to you in millimeter scale or whatever.
But when you say macrocosm, which is
I'm
a
talking
bigger
of a much bigger entity, which is made up of
microcosms, multiple
It
microcosms.
could be made
Okay.
up of multiple.
Okay. Thank you.
Yeah, thanks. So having seen some of these iconic microcosms, let's come to a microcosm that one of the microcosms, I'll be telling you about several that I work on in my own lab. This is a very iconic microcosm. And here is what I call the microcosm. You have the pollinating fig wasps, the female wasps, that are trying to enter this microcosm through the only single legitimate opening. This is a microcosm which will serve as a nursery for both seeds as well as wasps, which will develop within them. Just as you think of any structure which requires protection, here maybe a fort or a citadel, and you can think about life in a fort microcosm with all sorts of occupations and all sorts of, transactions happening. And there is weaponry also to defend or to, have offensive as well as defense. And you also have a gate which will come and shut down. So it's useful to think of analogies, at least in the fig microcosm that I will be telling you quite a bit about. So here are the microcosms that one of the microcosms I'm going to be talking about. This is Ficus racemosa, found even in Bombay. You will find it quite abundant. And the figs are borne on the stem. Each of them is a replicate of the processes. So I refer to each of these enclosed globular inflorescences. So the flowers are inside. There is only one gate or one legitimate entry port through which the pollinating fig wasp will enter. Yes? This is a group of students and postdocs over the years who have helped to contribute to all the information I will be presenting to you. Okay, so the microcosm gate, as I said, provides a one-way entry only, which is very common in Bombay, I can assure you. And in this process of entering, they lose their wings, the female pollinators, who also live only for 24 hours. They lose their wings, they lose their antennae in the process of burrowing in, which means that there is a high premium on correct microcosm identification. This is the only microcosm in which they are going to be able to lay their eggs. They can't very well come through and say, oops, I made a mistake, because they can't rectify any mistake. So this is a very stringent selection pressure, which it has placed on the fig and fig wasp system. Indeed, there are 800 species of figs in the world today, approximately, and each of them have their own species of pollinating fig wasp. So it's a very, very tight interaction. There are also, however, other ways to gain entry into the microcosm, and these are parasites of this mutualism between the fig and the pollinating fig wasp. So these are female gallers or parasitoid wasps who will lay their eggs either in the developing flowers, so that they also initiate a galling process, or they will lay their eggs on top of a larva which is already developing within the gall. So there are multiple layers of predator and prey interactions which are happening. If you take a section of the inner wall of this microcosm, you will see that there are seeds with the remnants of the stigma of the former flower, and then there are the developing wasps inside each of these galled flowers. Each one will develop, a single wasp will emerge from each of these flowers into which the wasps have laid their eggs.
Where does the pollen come from?
I'm telling you in a minute. So if you just follow this cycle, right? So you've got the early primordia, the early microcosm starting. In this particular species, the female flowers develop first. So wasps that have come from another microcosm, bear with me, we'll get to that point. So the female wasps bringing in the pollen come through the ostiole, which is the tight-fitting one where they lose antennae and lose their wings. And at this time, the female flowers are ready to be pollinated. They get pollinated, and in some flowers, you have eggs also being laid, which will develop into wasps. Okay, and then later you have the externally ovipositing wasps, which will come and parasitize this microcosm from the outside. Once this wasp development and seed development is complete, the male pollinators, whom I will introduce to you, I think in the next slide, they cut open another opening through this thick wall of the syconium, a rare example of males cooperating with each other. And they all cooperate because at that time, the male flowers are ready. The females collect pollen from the flowers, and that's when they exit. And then they come into another microcosm, which is at the right stage. So the whole cycle continues. Does that answer your question? Yeah, so they get the pollen later in development, and then they leave with the pollen from the microcosm.
Does this parasitic wasp lay only in those pods where there are a larva growing? Or
if they are parasitoids, they could also be galler wasps that will gall floral tissue to form a cavity within which their own larva. The pollinator is also a galler.
Okay. But do they sort of have a preference for those
Yes,
which are
yes.
already there?
I'm not giving you all those details because that will go in. But yes.
I'm fascinated at the behavior of the female,
Yes.
and it's I suppose it's programming, because it first visits a mature inflorescence, right, in search of potentially nectar or something.
No, they are non-feeding. They don't feed at all.
So they just visit it for what?
They come in because that's the only place where they will lay their eggs.
No, why do they visit the mature one where they pick up the pollen?
They are developing inside. It's a second generation.
Oh, so they mature inside.
They mature inside.
And they collect the pollen.
So the offspring of the females who come in, I told you the females live 24 hours. So they come in, lose their antennae, lose their wings, pollinate, lay their eggs, and die. That's it. End of the story. Their offspring, the female's offspring,
are born inside.
They are born inside this. They mate inside. Everything happens inside.
So why do the males cut open then? Why is that? What is that?
To let the females out. I'll show you the males.
My goodness.
I'll show you the males. Look at what the males look like. They are neotenic. They have a completely different developmental pathway compared to the females. The males are little, just a bag of genitalia, and sorry, and very strong mandibles only in the pollinator males, because only the pollinator males can do this cutting open process. So the others are all, in a sense, parasites of this cooperative activity, which further makes it very necessary for parasitoids to not lay eggs on males. As a matter of fact, male pollinators are rarely parasitized because they are necessary, and also the males are smaller and are not very good. I'll show you those details as we go on. Yeah. So if you just bear with me and bear with my story, I hope to take you through. So we've been fascinated with Ficus racemosa for a variety of reasons, and we made it one of the model systems in my lab because we have 3 genera, we have 7 species of wasps, which constitutes a good diversity. Not too few, not too many. Okay. The pollinator is a mutualist. The pollinator is a goaler. We have 3 other goalers, and we have 3 parasitoids. So we've got 3 carnivores, 4 herbivores, which makes a community of 7 species of wasps. So as I've told you, these are the Dramatis personae. This is the female pollinator and the females of the other wasps, which lay their eggs externally from the outside. This is the only one that gets inside. This is her male, and these are all the wingless neotenic males. So the males mate with their respective females and die within the microcosm. Okay. Now, we've been looking at what we call the oviposition windows, and the oviposition windows are quite very specific. So you have the goalers coming in first, and following the goalers, you have the parasitoids. The total duration of the gestation of a microcosm can be divided into a whole set of days. It's about 2 months, roughly. The largest is this interfloral period before the male flowers are emerging. This is the female flower period, and each wasp will come in for oviposition at a very, very specific time. And we know now the volatile compounds that attract these wasps to come in at very specific times. So this is a very important, sort of natural history of the microcosm. Okay, so if we are able to look at the whole community and we do survivorship curves, we know that gallers, and these are the ones that are the gallers, they are quite short-lived in their adult life. Okay, so this is a fig wasp adult survivorship. And you can see that the parasitoids live much longer. The pollinator, which is over here in black, lives only for 24 hours. Once it emerges from its gall, it has 24 hours and its adult life is over. So even amongst these members of this community, you have two main types of egg maturation strategies. One is pro-ovigenic, when the wasps eclose with all their eggs mature. So they are ready to lay all their eggs as soon as they eclose. We have another strategy that is synovigenic, where the wasps are maturing their eggs gradually over their 16 or 15-day period of their adult life. So naturally, the pro-ovigenic ones are what we call capital breeders. These do not feed as adults. So they have to use the capital that they developed when they were larvae. And the synovigenic ones are the income breeders. So they do feed, or they can feed. They have some amount of developed mouth parts. So in this community, then, we have a very nice partitioning by what are called life history strategies. We have really wasps on an R and K strategy continuum, from the very fast wasps that have a very short lifespan, smaller eggs, to the slow wasps that have the larger eggs, the fewer number of eggs also. So this really, I mean, we are very happy that we have such a lovely, nice community. So if we look at the developmental time of this microcosm, we have observed that the early arriving gallers tend to shorten the developmental time of the microcosm once they lay their eggs. And we think that this is because they start drawing nutrients very quickly. They form very large galls, which are very large nutrient sinks. The presence of seeds, the presence of pollinator galls are also shortening the developmental time. Here again, these are quite strong nutrient sinks, as I will show you in a minute. We think that this shortening of developmental time is actually to try and reduce the immobile, vulnerable stage within the microcosm, because the parasitoids want to lay their eggs on particular stages. And so the wasps want to hurry through those stages before they get parasitized. We have found that the parasitoids that come later want to lengthen the developmental time. And this is because they want to allow their progeny enough time to mature. Because remember, all wasps leave the microcosm at exactly the same time, when the male pollinator has opened up the syconium to allow the females of the pollinator, but all other females also leave at exactly the same time. So we think that there is then a trade-off between antagonistic factors, and we get an effect size of about 2 to 12 days, which is quite a large amount of time in a wasp adult lifespan. So there are pushes and pulls that are going on in development. So now, more recently, we are able to actually manipulate the occupants within the microcosm. We are able to produce microcosms that only have seeds. We are able to produce microcosms that only have pollinator galls. And we are able to have microcosms that are completely unmanipulated. We do this by excising the ovipositor in wasps that we introduce so that they are able to pollinate, but they are not able to oviposit. We are able to prevent the females from collecting pollen, and therefore they can only lay eggs, but they cannot pollinate, so they only produce galls. And with our manipulated microcosms now, and this is work in progress, we find that those syconia which have galls, so this is seeds and galls, this is galls only, this is seeds only, and this is the volume of the microcosm. You can clearly see that the presence of the galls is enabling the microcosm to garner more resources. So clearly, the presence of galls is affecting the resource allocation to these syconia.
Yeah, just one question.
Yes.
Hi, this is a very basic question, but in your initial slides, when you were talking about how the females lay the egg in a mature flower, stigma to pollinate basically and lay the eggs. So, of course, I want to ask, how do they know that the flower is mature? Do they visit a lot of flowers, or the mature one secretes some kind of fragrance? Or how do they know that the flower is the one that they have?
So the receptive fig has a very specific odor, which I will show you in a minute. Yeah. So every stage is producing its own bouquet of volatile organic compounds. And the wasps are sensitive to this bouquet. So that's how they come in.
Sorry, a quick one. I thought I kind of knew, but what is a gall? I
gall.
looked it up also. It wasn't clear. It kept telling me about livers and... or...
A gall is a modified flower in this case. So I go back, show you a picture of a gall. So if this is a flower with a stalk and then the stigma, the gall is where the flower no longer doesn't become a seed, but it becomes a nutrient chamber, which is supplying nutrients to the larva, which is developing. So the larva eats up the nucellus, eats up the floral tissue. So that's the gall. Okay? An occupied flower, which has now been taken over by the galler. Yeah. Okay. So that's actually pertinent because we come to. So I said about. So we've established that the syconium is larger when there are gallers present, which means that the gallers are changing the allocation to the syconium. And now we have looked at hormone contents of the galls, of the seeds, the galls, and the seeds and galls, syconium hormone content. And we can see very high levels of indole acetic acid and even of T-zeatin, which are plant hormones. So here we can, and this is actually more prevalent in the early phase of the syconium development. So clearly, and we are not sure yet whether the gallers themselves are mimicking and producing the plant hormones, or if the gallers are inducing the production of these plant hormones by the plants. Both are possible because we are now looking for genes for plant hormone production within the transcriptomes of these particular gallers. But clearly...
Renee, just quick question. How do you quantify these typically? The indole acetic acid, how did you extract it out? Or
we extract it and then we run an analysis.
Analysis of what kind? Like mass or
Mass.
mass? Okay.
Yeah.
Okay.
Absolutely. Yeah. If you just look at carbon and nitrogen masses and you compare a seed mass to this mass of a female pollinator, you can see that seeds, pollinators are much more expensive to make in terms of nitrogen as well as of carbon. But clearly, this is a mutualism. So it is a cost-benefit interaction. The plant is giving up good carbon and a lot of nitrogen to make these pollinators, because without these pollinators, obviously seeds will not be produced and the cycle will cease and will not happen. So to answer the question that was asked to me a little while ago, attraction to microcosms at all stages is chemically mediated. We have quantified the volatiles that are produced by the microcosms at all these stages. And you can see that early on, I'm just depicting each volatile as one color. You see earlier on, there are many, many more volatiles, whereas later on, the volatile profile becomes more simple, and it's mostly esters and alcohols in the fruit dispersal stage. And we get about 55 volatile organic compounds from all these stages. We've also done heat maps and looked at the volatilome across the different stages, and we find there are some co-expressed modules of volatiles, and we have some ideas as to why certain volatiles are co-expressed at particular phases in the development of the microcosm. Now, as I said, there are 800 species of figs, and each one has its own unique pollinator species. So in this messy environment where many of the compounds are also quite similar, this is sort of the chemical equivalent of what auditory biologists or acoustic scientists refer to as the cocktail party effect. You've got a lot of noise, but you have to pick out your signal from the noise. And we have resorted to multidimensional analysis to show that sympatric fig species that are fruiting and flowering in the same area, have very distinct volatile profiles at the flower receptive stage when the wasps are coming in order to lay their eggs and pollinate. So clearly, even on a statistical way, they are quite separate. Yes.
So the wasps get attracted, and after the female gets in, does the profile of the volatile change? Or
Yes,
would
it
it
does.
just change naturally anyway? Does the wasp contribute to the change, or does it happen?
If the wasps do not enter and do not pollinate, or the figs will be aborted.
I see.
Because the tree does not want to have a sterile syconium developing. So the tree is also assessing if the wasps excessively lay eggs also, the syconium will be aborted. So this progression that you see is a result of all the development of the wasps and the seeds. Does that answer your question? Okay.
Renee, could you explain me these multidimensional analysis? What are these dimensions and how did you do it?
So these are very simple. I mean, what we use, you can use a principal component analysis.
On which data are you looking at?
On the profiles, the volatile data.
Volatile data, okay.
Right?
Okay.
So you've got many, many individuals of one species, and you collect the volatiles in situ.
Okay.
Right?
And these are the two principal components that you're plotting.
These are the two principal.
No issues. Thank you.
Yeah. Okay. So clearly, fig wasps, it even, I mean, in a statistical way, you can see the differences in their profiles. Now, we've also gone on to look at olfactory receptor genes. And just to point out that this is our species, the pollinator of Ficus racemosa. It has only 56 olfactory receptor genes that we've annotated. Compare that to 350 in an ant species, which is a generalist, and so on. So there is a massive reduction in olfactory receptor genes. We also have ectopic expression of these olfactory receptor genes, and recently more and more people are beginning to report ectopic expression. And here you can see that we've got ectopic expression. I mean, of course, most of the expression in antennae, but we also have expression in other parts of the body. And I also point out to the ovipositor. So we've been able to show that the ovipositor is capable of olfaction with other behavioral experiments that I will show you in a minute. Gosh, I'm running out of time. Okay, so in this microcosm, you have eavesdroppers on the microcosm scent. And here you have ant species that are converging onto the syconium when the pollinators are arriving in large numbers to enter the microcosm. So here you have the notorious weaver ant that is snacking on the pollinators that are trying to push their way into this microcosm. So we've done behavioral analyses, and we have shown that these odors, so we've only tested with the odors, and we have shown that these ants are sensitive only to the odors of the stage, which is the B stage. So what I have over here are the different stages of the development of the microcosm. The B stage is when the odors are being produced to attract the pollinators in. And you can see that these two species of ants are only attracted to the odors of the B stage, and also in a couple of cases to the odors of the D stage when the wasps are leaving the syconium. So these are 2 points when you have massive numbers of wasps, 1 point when the wasps are coming in, and the other point when the wasps are leaving. So we've also been able to show that this response to fig-associated odors is a learned response. So when we tested on naive ants that had no former opportunity to learn the association between the odor of a B receptive phase fig, and we test both species of these ants, the naive ants, we find that there is absolutely no response. So this was one of the first cases where we showed that ants can learn odors and make associations with them. So some other occupants of this microcosm. We have a thriving community of nematodes living in this microcosm. This is a very ancient association. These are examples of nematodes coming out of the abdomen of a fig preserved in, millions of year-old Dominican amber. And what we realize, you have a question?
Yeah, just sorry. So you said that a fig that doesn't get pollinated aborts or overpollinate aborts. So that means all the figs we eat have all this wasp thing happening inside them. And now you're adding nematodes, and actually, what are we eating?
So the commercial figs are parthenocarpic. Okay? But, if you eat figs in the Mediterranean region, you will get wasps in them. Okay, but if you eat figs from Egypt, from Saudi Arabia, from all those places, they are parthenocarpic.
What does that mean? I'm sorry, I don't know. What does that mean?
Parthenocarpic is producing seeds without pollination.
Okay.
So you get the crunchiness, but you don't get the. But the French are very particular about their figs, and so they have fig wasps in it. But that's good protein in any case.
It adds to the taste.
It adds to the taste.
Okay.
And the crunch. I agree. Okay, so nematodes. Yes, nematodes are fabulous. So these nematodes are hitching rides on the fig wasps in order to enter the microcosms. They are highly specialized. So just as you have species-specific fig wasps, you have fig wasps and fig species-specific nematodes. Nematodes do not cross the species boundaries, okay? So Phorese, which is a technical term for whether you are a rider or you are somebody that's offering a ride, there are lots of things that need to work for Phorese to be effective. And the most important thing is that the life histories of the riders as well as the rides need to be synchronized. Yes. I recently did a review on this. So if you're interested, you're welcome to. Okay. So let's look at nematodes and why are nematodes also so successful? They're successful because they have this very important dour life history stage. And in this dour life history stage, it is a sort of a resistant stage where the nematode is not just going through the whole life cycle as usual, but it is an exploratory stage, or it is a stage when the nematode is not unsure about the conditions within the microcosm or within the Petri plate or whichever, and the nematode is sort of preparing for dealing with stressful conditions. So this is, I've just taken from the C. elegans life history, but indeed the dour life history stage is very, very important. They are kind of hibernating or stasis. So they are not doing any, they're not going through the whole cycle. They're just arrested.
Yes. Okay, stasis. Okay.
Yeah. So in our,
as opposed to just being unhappy and upset.
Yeah.
Down.
So in our system, in Ficus racemosa, we've got 3 genera. We've got Schistoncus, which is a plant parasite. We put it as putative, but it's pretty sure it is. And here, all our nematodes have separate sexes. If you remember C. elegans, it can be hermaphrodite, right? Or it can be female, right? But all of our fig nematodes, at least the ones we deal with, have separate sexes. We have Schistoncus, which is a plant parasite, Teratodiplogaster, an animal parasite, and Pristioncus, which is not so common in our system. So we decided only to work on these 2. And I'm not showing you results from this experiment, but what we were able to establish with a whole battery of behavioral assays, nematodes hitchhike only on the female pollinators. So you give them a choice of male and female pollinator, they choose the female. You give them a choice only of volatiles by making slits in the agar so that, only volatile cues can diffuse. And these are experiments we do with single nematodes. Normally, people put a bunch of nematodes in and then do tests. But we have developed an assay system where we can use single nematodes for this. We can take extracts of cuticular hydrocarbons, and again, the nematodes choose. And we give them cafeteria experiments, and again, the nematodes only hitchhike on the female pollinators. We've also established that every fig wasp species and sex has its own unique cuticular hydrocarbon signature. So we think that the nematodes are using a combination of volatiles, which they certainly can, as well as cuticular hydrocarbons to make their choice. Why are they only choosing to hitchhike on female pollinators? Because it's the female pollinators that enter the syconium. The others, the males don't. And the others are only entering the syconium through their ovipositor. And it's a very thin ovipositor, so we have no records of nematodes coming through the ovipositor. Yeah. So here is the fig wasp nematode interaction. I have shown you this before. You've got the developing syconium. You've got nematodes jumping onto female pollinators. I don't have a video here. Now I remember I should have put it in. They do a beautiful behavior called nictation. So they stand on their tails and they wave their front ends where there is the olfactory receptors, and that's how they make their decision, and they jump onto the female pollinators. The female pollinators then carry the load of nematodes, and then they come into the receptive stage. The nematodes leave the body of the pollinator and the whole cycle continues. So we were interested in the impact, of course, of the nematodes on this whole business. And we find that the animal parasitic nematode impacts the flight. So I'll show you a flight assay in a couple of slides from now. So those wasps that are infested with the teratodiplogaster, they are the ones that affect their vehicles so strongly that the vehicles have a very, very short flight duration in the assay. Whereas if you take a control where we just use the normal number of nematodes present, we don't get, it's equivalent to what's happening in a low number of Schisthoncus or teratodiplogaster. We've done assays on predation risk on the pollinator wasp, because you've seen that the ants attack the pollinators as they are entering the syconium. Wasps that are carrying a heavier load of nematodes take much longer to enter this osteole, and therefore they are more prone to predation risk. We've measured the time taken to enter the osteole, and you can see that with the heavier load of the parasitic, the animal parasitic nematode, the time taken is very, very much longer.
Sorry, I keep asking. I'm just very intrigued.
Yes.
So what is the weight of 30 nematodes with reference to the weight of the wasp? I mean, just to get a sense of what this thing is carrying.
I don't have an image here, but I would say a wasp is about 2 mm. And we can back calculate. I would say 30 nematodes would be about 30% of the weight of us. Yeah. Okay, so here too, we could, sorry. I have to hurry. We have also looked at lifetime reproductive success of the pollinator wasps that are carrying these nematodes. How do we measure these lifetime reproductive success because we allow single wasps to enter, and then we count the number of offspring that they produce. And you can see that the animal parasitic nematodes reduce the lifetime reproductive success of the wasps significantly. We looked at microcosm growth, and we have been able to demonstrate that the nematodes also reduce the growth of the microcosm in the sense that the wasps that are laying eggs and galling are apparently not as efficient at doing their job when they have been parasitized by nematodes. We've looked at the seed set in those microcosms with wasps that have carrying nematodes, and they have to do the pollination. And once again, we see a dramatic decline in the numbers of seeds set by pollinators that are infested with nematodes. So the bottom line, if we are seeing all these effects, why are nematodes still in the system? It's quite interesting, but I want to go back to this slide and show you that we've done our tests with increasing numbers of nematodes. We've taken the maximum number of nematodes ever found on a wasp, and we've designed our tests accordingly. But if you look at the wasps that are dispersing, so I call it embarkation and disembarkation. So the nematodes are embarking on the dispersing wasps. You can see that a lot of them do try to get on. So you've got a frequency distribution with a longer tail. But if you look at the arriving wasps, the wasps that come back to a bee-face fig, you can see that most of them have very few numbers of nematodes, which tells us that there is a very, very strong selection for fewer wasps to actually carry nematodes and fewer nematodes to be tolerated by the wasps. Now, we've done experiments also to show you, to talk about these nematodes have separate sexes. So if sometimes there can be only a single female wasp that will enter a syconium. If she's the only one to enter a syconium, and there is only one sex of nematode present, obviously it is game over for the nematode. So we've done tests, and we have shown that some nematodes, the Teratodiplogaster, for example, will not get on if we offer it a wasp which is devoid of nematodes. The other species prefers wasps which have no nematodes, but this species gets on in pairs. So we've got very nice videos, which unfortunately I don't have to show you right now, that these tend to jump on in pairs, and we have no idea how they are able to figure out which is male, going to become male, and which is going to become female. 5 minutes. Okay, I'm rushing. Yeah. So just to tell you a few more interesting stories only on the fig thing. So remember, I told you wasps are ovipositing from the outside, but once again, because these wasps can only mate within the fig, they have to ensure that there will also be mates for their offspring. So this is a very, very peculiar game. And because, as I've said before, they need to oviposit preferentially into females rather than into males, they also have to be able to sex the prey from the outside through the ovipositor. So we've also looked at the ovipositor of the different species of wasps. We've also looked at the amount of force required to penetrate through the Syconium wall, but that's a whole nother story. As I said, we've been able to show using a modified electroantennogram on the ovipositor, something we call an electro-ovipositogram. And we've been able to show that the ovipositor reacts and is sensitive to fig wasp and fig volatiles. So antennae are not the only olfactory organs. Wasps sometimes aggregate on the surface, and we've shown that they leave footprints on the surface. With the footprints, and we've been able to characterize the footprints, they are probably able to tell if other wasps have oviposited into this closed microcosm. Okay, so the last part of my story, which is, I think, very interesting, is about dispersal of the fig wasps. We know that the wasps are wind dispersed, but they also are capable of more directed flight to their targets. So in order to infer their flight abilities, we've done tethered flight analysis using an optical tachometer. We get data and fast Fourier transform them. So every wing beat we are able to calculate. We also do lipid estimation on the amount of lipid in the somatic modules, which can fuel flight as opposed to the reproductive modules. We've measured their metabolic rates of the different wasp species. And just as I said, we have a fast and slow continuum with life histories, which I showed you earlier. We also show that fast wasps are in a great hurry to reach microcosms before they die, because the fast wasps live for shorter and the slow wasps live for longer, as I've already told you. And this bears up in our flight duration, somatic lipid, and all the other analyses. The last bit of data. So we had these data for our species, Ficus racemosa, and the wasps. So we wanted to compare it with data from another ficus species, Ficus hispida. These Ficus hispida trees have a closer spatial distribution. The Ficus racemosa trees are much further apart. So wasps that need to get from one tree to another would have to need much more fuel. And you can see the difference between racemosa and hispida in the lipid stores, in the flight duration compared to hispida. So this beautifully ties up with the fact that you have the petrol and the gas that you need to get you from one tree to another. So I am going to wrap up by saying I've showed you a whole diversity of interactions. And there are many, many fixed species with all sorts of wonderful things in them. So I'll end there. Thank you very much.
Thank you very much. Perfectly on time. Thank you, Renee. Yes. So there are lots of questions. So let's first start with the physicist, Rameez.
Hi. So I'll start with the question that Jyotishman really asked, like the scale that defines them. And you said that it's not the scale, it's that it's self-contained. But in some sense, the entire rest of the talk was about how it is not self-contained, right? Like things are coming in.
Come in and things go out. So you can think of a completely, I mean, in that sense, all ecosystems are not entirely closed, right? I mean, there's always some amount of gas exchange.
So, yeah, I thought I'd ask, like, are all fruits microcosms in some sense, or?
For a dipteran larva that is growing inside. So if you eat a guava and you open it and there are these wiggly maggots in it,
Okay.
It's a microcosm for the development of the dipteran larva, right?
Sure.
Sometimes you eat, sometimes you don't. If you open them and there are too many maggots, you throw it out otherwise. So it depends on how you, for what purpose are you defining? if I can just go back one slide, sorry. So we've modeled this to show the minimum number of trees required in Ficus racemosa to sustain all members of the community. Because I've shown you that different wasps have different requirements, and they need to find their prey at the right stage, which I haven't shown you all those data. So we've been able to model how many trees would you need in order to sustain the entire community of wasps. Does that, yeah?
Okay. Yeah, so this fig, it is available for a whole year?
Very good question. Yes, in order to sustain the wasps, they're the only group of plants that are fruiting and flowering continually. So an individual will have a gap phase, but another individual will be flowering, fruiting. So the wasps will move between the trees. That's how the system is maintained. Yeah?
Why do all the wasps leave the microcosm at once?
Oh, if they don't leave at once, the composition, the seeds have to be dispersed, right? So very quickly, the composition changes to alcohol esters is the ripening. And if they don't leave, then the birds and the monkeys will eat them, and then the wasps will land in the bird and monkeys' digestive system. So they have to synchronize their development. All 7 species have to synchronize their development and go at one time.
Hi, Renee, great talk. I have a 2-part question.
Yes.
One, are there examples of systems where there are multiple wasps or multiple species competing for the fig? And if yes or no, or does that mean that these are very stable evolutionary outcomes?
Okay, so I gave you the textbook story, and our system does follow this textbook story. But on many island systems, because islands, as you know, are highly fragmented systems, prone to fluctuations. So on many island systems, you have pollinator overlaps. Now, you have to remember that because the development of the wasp has to keep tandem with the development of the seed, and the size of, if I showed you, you can have fig species that are 4 millimeters in size, and you can have a fig fruit which is 2 inches or whatever. Obviously, the flowers in this one will be much bigger than the flowers in that one. So even the resources for the pollinators have to match. So every pollinator cannot, get into another fig. That's why there is a very tight selection, separation. Okay, so only closely related fig species sometimes share pollinators. That answer your question? Okay.
Thanks. This was really informative. Now, we have a lot of fig trees around here. There's one right behind the Vishnu statue. The colony is full of them. And there are lots of birds that come. I mean, it's a tree which is full of hornbills, for example. So now I'm just wondering, after what you said, are they coming for the fruit, or are they coming for these wasps and nematodes and ants and whoever else? So are the birds coming because of the fruit or because of all the other people who are on it?
Well, contrary to a popular conception, birds have a very good olfactory sense. And I haven't shown you data, but we've looked at bird dispersed fig odors and mammal dispersed fig odors, and they are very different. So the figs that you are talking about here have an odor that is largely attractive to mammals, but will also be somewhat attractive to birds. But the birds are coming for the ripened odor. So they will not come to odors of an intermediate, Does that answer your question? Bats, yeah. So it's more of a bat odor.
So,
Yeah.
Hi. So when you were talking about nematode loads on the wasps and the correlations, you talked about finding a wasp that had a 30 plus nematodes, and that was sort of the maximum that you discovered. I'm assuming that that was in situ. Why is it that certain wasps are more susceptible to nematode attack?
I don't know the answer to that question. The way we do these experiments is we tether wasps and then we count a certain number of nematodes, and then we allow the nematodes. So the nematodes enter through the spiracles, okay? Sometimes they get in through the intersegmental membranes, yeah? And we really do not know whether the wasps can somehow protect themselves from getting overloaded with nematodes. That's an interesting question, and we don't have the answer to that.
Okay, so I would like to know if I have two figs, one has a small microcosm inside it and the other is not there. Is it possible to know just by looking from outside how ripe it is, that which one has wasps and ants in it and which one doesn't? I would be very interested in knowing.
From
if there
the
is a way.
outside,
Yeah.
that would be difficult unless you do some soft X-ray or something.
Okay. not by looking how ripe it is. Or...
See, with the figs that we work on, we are now very, we have other players that I haven't talked about because I wanted to keep it. We've got some aphids, we've got other types of, players in this game. But no, you can't just look at it and say what is inside. That's the beauty of this business.
Hi, here. Hi, here. I'm here.
Yeah, hi.
Sorry. So in extension of the nematode thing, I just want to ask, so you just explain the way that the nematode hops on kind of on the wasp and then it gets in. So now the way I see it now, the wasp is itself laying the eggs and it'll have its life cycle where they'll grow. So once they enter, the nematode enter, is it in the dower stage? And then when the females have left, then it now starts to continue its own life cycle? Is it the way it is, or it's something different? Like, how do nematodes survive there? Are they constantly?
The nematodes hop onto the females. In 24 hours, if the nematodes are lucky, they will get into another fig. They can't guarantee it. If they overload the wasp, it's less likely that that wasp will successfully make it inside another fig. Once the female enters, then the nematodes come out of the abdomen of the female, then they continue their development, then they mate, lay their eggs, and the cycle of the nematode continues inside.
So my question here is, now the female that is lay the eggs in the that fruit, are these two life cycles parallel, or is one waiting for the other to finish and then it'll start?
Which life? So the wasp life cycle is going on.
Yes.
The nematode is matching its life cycle with that of its ride, because it's a rider. It has to match its life cycle with that of the ride.
Inside the fruit itself.
Right, absolutely. So that when the wasps are getting ready to come out, they have to wait for the wasps. It's fascinating because you actually see them waving, standing on their tails and waving, and then jumping onto the wasps.
Okay. I have to close this session here. I'm sorry, but let me thank Renee once again. And there are multiple questions, Renee. Let's have a big round of applause. There are multiple questions that you have sort of instigated. So we will have a tea and snack session just after out of the candy, and you can catch hold of her, including Maitri. Maitri had a question. I'm sorry, I had to cut that. But we will have more questions as we walk out.
Sure.
Thank you for coming in today in large numbers. And sort of 5:45, I think again there's a session that's going to start. So please, yeah. Huh? Oh yeah, and we don't have Anjeer Barfi today, but we have Kaju Barfi, I think. So yeah. So.